Good day, ma'am. Our lesson entitled Midterm Laboratory Exercise DNA Analysis. Before we proceed to our lesson, let's start with the lesson objectives and slide contents. Lesson objectives. Students should be able to explain the importance of DNA analysis and enumerate the steps of DNA analysis. Contents. Slide 1. Title, objectives, slide contents, and presenter's identity. Slide 2. Description of DNA, location, composition, and nature. Slide 3. Description of DNA analysis and how it is used to solve crimes. Slide 4. 4 steps in DNA analysis. And slide 5. 4 types of DNA evidence analysis. Now I will discuss the DNA or deoxyribonucleic acid, a self-replicating material which is present in nearly all living organisms as the main constituent of chromosomes. It is the carrier of genetic information. DNA is made up of molecules called nucleotides. Each nucleotides contains a phosphate group, a sugar group, and a nitrogen base. The 4 types of nitrogen base are adenine, thymine, guanine, and cytosine. The order of these bases is what determines DNA instructions or genetic code. Forensic DNA analysis is the use of DNA specimens in legal proceeding. Just as people can leave fingerprints when they touch a surface, so too they can leave biological materials that contains DNA. DNA is generally used to solve crimes in one of 2 ways. One is in cases where a suspect is identified, a sample of that person's DNA can be compared to evidence from the crime scene. The result of this comparison may help establish whether the suspect committed the crime. Second, in cases where a suspect has not yet been identified. Biological evidence from the crime scene can be analyzed and compared to the offender's profiles in DNA database to help identify the perpetrator. Now, the steps in DNA analysis. Several basic steps are performed during DNA testing regardless of the type of test being done. The general procedure includes 1. The isolation of the DNA from an evidence sample containing DNA of unknown origin, and generally, at a later time, the isolation of DNA from a sample. Second, the processing of the DNA so that the test result may be obtained. Third, the determination of the variation in the DNA test results from a specific region of the DNA. Fourth, the comparison and interpretation of the test result from the unknown and known samples to determine whether the known individual is not a source of the DNA or is included as a possible source of the DNA. Now the last slides, types of DNA evidence analysis. Polymerase chain reaction. The copying process, known as polymerase chain reaction, uses an enzymes to replicate DNA regions in a test tubes. By repeating the copying process, a small number of DNA molecules can be reliably increased up to billions within several hours. Second, short tandem repeat is a forensic analysis that evaluates specific regions or loci that are found on nuclear DNA. The variable polymorphic nature of the STR regions that are analyzed for forensic testing. Third, Y chromosomes. Markers target only the male fractions of a biological sample. This technique can be very valuable if the laboratory detects complex mixtures within a biological evidence sample. Because the Y chromosomes is transmitted directly from a father to all of his sons. It can also to trace family relationship among males. And the last one, mitochondrial DNA. These analyses allow forensic laboratories to develop DNA profiles from evidence that may not be suitable for STR analysis. Mitochondrial DNA technology analyze DNA found in a different part of the cell, the mitochondrion, such as hair shafts, bones, and teeth. And that would be all for this midterm laboratory exercise DNA analysis.